Mismatch repair mechanism in eukaryotes. With the entire mechanism being a highly conserved mechanism and its ability to maintain genomic stability, uh, the mismatch repair in eukaryotes too look at or address what is called as single base pair mismatches or short insertions and deletions that are associated with the process of replication. And for the mismatch repair, it is a challenge that it has to be the mismatch or the lesion has to be repaired before the DNA replication completes. So let us look at the learning outcomes of the session. During DNA replication in eukaryotes, the errors in the nucleotide incorporation is just on an average about 1 in 10 raised to 4 or 1 in 10 raised to 5 nucleotides. And this can lead to, if not corrected, this can lead to genomic instability. And of course, it depends a lot on the locus of the mismatch and also the type of the mismatch that the genomic instability is associated. The mismatch repair machinery works closely with the replication machinery to ensure the fidelity of the replication mechanism. So we already know that DNA polymerases with its 3' 5' exonuclease activity is already able to proofread while carrying out replication. But yet, if in the process of replication, if it is unable to identify a wrong nucleotide that it has incorporated and moves ahead with replication, then such mistakes have to be corrected. And those mistakes are what is corrected by the mismatch repair mechanism. In fact, what has been observed is that if there is a compromised MMR system in eukaryotes, then it is associated with cancer. The challenge that the mismatch repair system has, as we just mentioned, is to repair the errors before the replication is complete and to make the correction in the newly replicated strand itself. It cannot correct the template. It should not correct or change anything in the template. It has to correct only the wrong nucleotide that has been incorporated in the newly synthesized DNA. And therefore, that is a big challenge for the mismatch repair system. This mechanism, too, is the manifestation of DNA protein interactions and conformational changes. Importantly, it prevents mismatches from becoming a permanent mutation. So, just getting to understand MMR in eukaryotes, as in the steps involved, the first step, of course, is in identifying the mismatch based repair or in identifying the indels, that is, the insertion and the deletions. Identify the new strand. Okay, so it just does not have to identify the mismatch based repair, but having identified the mismatch based repair base pair, it also has to ensure that it identifies the strand that contains the wrong nucleotide, and that is done by looking at the discontinuity in the newly synthesized strand, because we all know that DNA replication is semi-discontinuous. So there are Okazaki fragments present in both the DNA templates, and therefore you would have discontinuity. And those discontinuities are basically recognized by MMR as or that becomes the basis of recognizing the newly synthesized DNA. So the identification of the new strand or the newly replicated strand is also taken care of by the MLH molecule, that is mut L homologue. The excision is done generally from the 5' end of the mismatch because the exonuclease present is 5' to 3' exonuclease. However, the incision or the nix can be on either side of the mismatch, and hence in eukaryotes you can have distinct 5' MMR and 3' MMRs. Now, having carried out the excision, there remains a gap. So the gap is then filled up by the DNA polymerase pCNA FEN1 DNA ligase complex machinery, which is observed, which is part of the DNA replication machinery, and that's the reason why one associates the mismatch based repair system with the DNA replication system itself. What has been observed is that both DNA pol epsilon and DNA pol delta are involved in the MMR process. Now we will look at the molecular system or the molecular machinery which is responsible for identifying the mismatch. So that in eukaryotes is uh, homologous to the mute S of the prokaryotic system, and therefore it is called as mute S homologs. But interesting to note is that the mute S homolog is a heterodimeric homolog. That means in the prokaryotic system it is a homodimer, but in the eukaryotes it's a heterodimer. It recognizes the mismatches and it is related to the AAA plus family of ATPases, just like the mutes of the prokaryotic system being associated or related to the ABC family. So when you look at the structure and compare it with the prokaryotic mute S, and you will find that there are two MSH complexes present in whether you look at Saccharomyces cerevisiae or you look at humans. This over here is humans. So you have two basic mute S homologs. One is what is called as mute S alpha, and the other one is what is called as a mute S beta. So the mutase alpha comprises two monomers, one which is MSH2 and the other one is MSH6. So the MSH2 MSH6 heterodimer comprises the mutase alpha, and the MSH2 MSH3 comprises the human mutase beta. Now, what is interesting to note is that they have lots of structural and functional similarity to the mutase of the prokaryotic system. If you look at the prokaryotic system mutase, you will find that you have two monomers completely identical. So they are structurally they are the same, identical, but functionally they differ. So they have what is called as a functional asymmetry. Now, when you look at the structure of the mutase in the prokaryotes, there are 5 domains. Okay, the domain 1 is the one that is the mismatch binding domain. So it is able to recognize the mismatch and bind to the mismatch of the DNA. So this is where you have both the monomers binding together to the mismatch, and therefore when it binds to this DNA and binds to the mismatch, there will be a conformational change at this in this domain, which through domain 3 and 4 will propagate to domain 5, and this domain 5 is basically a composite dimeric structure. So here you have a helix turn helix motif which is able to change the conformation once you have the change of conformation in the mismatch binding domain. And because of this conformational change, what also happens is the inherent ATPase activity of the domain gets activated. Plus, it also requires the binding of the mutel. So the binding of the mutel is at the domain 2. So when you have the binding of the mutel to the mutel 2, you have ATPase activity enhanced further. Also, what is interesting to note is that the ATP binding itself to this domain is able to bring out a conformational change. And these conformational changes are important in progression of the mismatch mechanism repair system. Now, in case of the human or the eukaryotic mute S homologs as well, what you observe is that you have the uh, mismatch binding domain over here. You also have the domain 2 which can bind to the mutel homolog, and you have of course 3 and the 4 domain which are primarily made of helices, and you will find that any conformational change in the mismatch binding domain will be reflected finally in the domain 5 which has inherent ATPase activity. And you can see over here that it is at the domain 5 region itself that you have the dimerization domain. So this is where you have the dimerization of the two monomers, not the two monomers happening, but the monomers over here are different. They are not identical. So you have one as MSH2 and the other one as MSH3. Here you have MSH2 and this is MSH6. There are MSH4 and MSH5, but they are not involved in mismatch repair. They're involved in other molecular mechanisms within the cell. Now, interestingly, uh, what is very important to note is that you have the MMR repairing both mismatches as well as indels. So the mutex alpha is able to recognize mismatches, that is single base pair mismatches, and 1 to 2 nucleotide insertions or deletions. But the human mute or even the yeast mute S beta is able to recognize 1 to 15 nucleotide insertions and deletions. So lesions that are because of insertions and deletions are also recognized by the MMR or the MSH homologs. A point to be noted over here is that the MSH specifically is able to identify distortions that happen in the DNA that could be because of a mismatch, but they confirm whether it is a mismatch or a not. So it's not just identifying the mismatches, but also kind of confirming whether it is a mismatch or not. So when you have a mismatch in the DNA, this A has been added by the DNA polymerase that has moved ahead without correcting the wrong nucleotide that it has added. In such a case, what is observed is that you would have the mutex homolog coming and attaching itself to this. And on attaching itself to this, it creates a kink in the DNA. Now, the kink in the DNA is only to ensure that you have the motif, that is the phenylalanine 436X glutamate 435 34 motif of the mismatch binding domain interact with the mismatch. So the conserved phenylalanine 436, residue 436, is able to stack within the mismatch, okay, through the minor groove of the mismatch, okay, and once it stacks within the mismatch, so because there is no concrete hydrogen bonding between the mismatch base pair, it is able to stack in, and after stacking in, the glutamate 434, which is also conserved, is able to hydrogen bond with the two with the mismatch a base pair that is there. Now, on doing so, the bent DNA again becomes unbent. But then what has happened is that the interaction of the mutex homolog has become stronger because of the interaction that the phenylalanine and the glutamate is making with the DNA at the mismatch. So you can see very clearly that this is a protein DNA interaction that enables identifying and confirming. One other thing that the mutex homologs have been reportedly observed to be doing is flip out the wrong nucleotide so that it is basically ensuring that yes, it is the mismatch. So having done so, okay, what is observed is that the conformational change in the mismatch binding domain because of the interaction of the motif with the mismatch uh, brings in so there's a conformational change because of that, because there's a conformational change that radiates to the domain 5, and domain 5 is nothing but the nucleotide binding domain. So what happens is there is an exchange of ADP with ATP, so there is a nucleotide switch that is happening, and the moment there is a nucleotide switch happening, the conformation of the mutex H homolog becomes such that it becomes a sliding clamp. Now, because it becomes a sliding clamp, it can slide either towards this direction of the mismatch or towards this direction of the mismatch. Now, what is interesting also to notice that with this conformational change, okay, it is basically one of the MSH that binds to the DNA, and the other MSH is free to bind to mute LH or the MLH, the mute LH L homolog, that is the MLH. And this MLH MSH complex formation is very important in the MMR system. So this is something that is important to note, that not just the conformational change brings in such a change that it becomes a sliding clamp, but it also enables the mu-tail-H to be recruited or to be bound to the MSH. So that is something that is very important. And in fact, the MLH binding to MSH DNA complex ensures that the affinity with which the MSH binds to the DNA is enhanced. So basically, effectively, the MLH is also ensuring that the MSH binds to the MLH. Okay, and therefore, this important MLH MSH complex formation has intermittently led to the fact that MLH then acts as an endonuclease, which we will just look at. So here, the ATPase activity of the domain 5 also gets active, and when it gets active, you have the ATP getting converted to ADP again. So the energy that is obtained from the hydrolysis of the ATP is understood to help in the sliding process. Now, how does the MLH basically work, and what is its structure? So when you look at the E. coli mute L, okay, um, okay, in the E. coli you have what is called as the mute H as well, and the mute L is the molecule that binds to the DNA after binding to mute S and recruits mute H, which is an endonuclease. But there are many bacterial systems which do not have mute H, and in such a scenario, the mute L itself acts as an endonuclease. The same thing has been observed in eukaryotic system as well. And when you look at the structure, compare the structure of the mute L in the E. coli or in the prokaryotic system with the mute L homologs in the eukaryotes, you will find there are a lot of structural similarities. So let's consider first the dimer of the E. coli, which is generally again a homodimer in case of the prokaryotic system. So it can exist in two conformations, that is the open conformation and the closed conformation. So primarily it is the C-terminal domain which is the dimerization point. So you have even the open complex bound to each other at the C-terminal. So this is the dimerization domain. Now, what is observed is that on binding of ATP to the N-terminal domain, the N-terminal domain dimerizes. So this is something very important. And this happens when you have the mute-S binding to the mute-L. So therefore, an open conformation gets closed. And when it gets closed, you can see that there is a groove formed that allows it to bind to the DNA helix. So you can have this groove or this gap that is there between the N-terminal domain and the C-terminal domain. And the linking of the N-terminal domain with the C-terminal domain is through linkers, and at the center you will have a gap or you will have a groove that allows the DNA, double-stranded DNA, to pass through. So it's effectively what we understand is that you have the mute alpha, mute L absolutely wound around the double-stranded DNA. So that is what is understood. Now when you look at the eukaryotic system, this is the yeast mute-L homolog, and this is the human mute-L homolog. And one observes that in the eukaryotic system, they are not homodimers, they are heterodimers. So one of the monomer is the MLH1 monomer and the other one is the PMS1. The same you would observe in case of the humans as well. Now, again, therefore, what is important to notice that at the C-terminal domain, you would have the interaction such that the dimerization primarily begins at the C-terminal domain. The N-terminal domain has a dimerization domain, but it gets dimerized only when ATP comes and binds to the N-terminal domain. And so when it comes and binds, that is ATP comes and binds to the N-terminal domain, you have it dimerizing and closing to have again a groove that allows the DNA helix to pass through. So you can see how similar structurally they are, although the composition and the structurally what you are very clear about is that the two monomers that is forming the mute L homolog is not a single protein, it is two different proteins. So it's a heterodimer. So this is important, the formation of the DNA binding groove, and that happens only when the dimerization at the NTD happens, so the N-terminal domain happens. The MSH binds to the MLH monomer of the MLH homolog. So that is important. Now, another interesting fact is that the MLH molecular system is very closely related to another family, which is the GHKL family. Under the GHKL family belongs gyrases, HSP90, histidine kinases, etc. Now, what they share, what are these molecules of the superfamily GHKL family have? They have shared conserved motifs in the ATPase domain. So this domain you will find they are similar, and that is why they are also related to the GHKL family. So what has been observed is there are a lot of structural conformational changes that happen in the GHKL family, happen even with the mute L homolog. So before we go to looking at what are the structural implications of belonging to the GHKL family, it is important to note that there are three MLH homologs. That is, you have mute L alpha, you have mute L beta, and you have mute L gamma. But of the three, it is the mute L alpha which is closely related to the MMR system, because even if the percentage of mute L beta and mute L gamma in the cell decreases, it is not affecting the MMR system. But if mute L alpha is defective or is not working properly, then the MMR system is known to be affected. Now, when you look at the mute L with relation to it belonging to the GHKL family, you will observe that, like observed, only when the ATP binds to the ATP binding domain at the N-terminal domain, you will immediately have dimerization over here, and from this position it compacts to form a structure like that. When it compacts to form a structure like that, the ATPase activity of the N-terminal domain becomes active. It hydrolyzes the ATP, forms ADP, and the ADP is removed to come back to the source. So this dimerization cycle keeps on going on, which is very similar to how it is with HSP90. So that is something that has been observed, and that's the reason why it is related to the GHKL family. Now, when we look at the C-terminal part of the MLH, so here you have the MLH1 and here you have the PMS1. So what is interesting to note is this, that this part of the PMS1 has a very conserved motif which is responsible for attributing the MLH with endonuclease activity. And interestingly, uh, this portion, or rather the MLH1 and the PMS1 together, are associated with metal ions. So zinc is present, which is associated with the cysteine residue of the MLH1, and you have Mn plus, which is basically associated with the endonuclease motif, and therefore the zinc and the Mn have different functionalities. Mn plus has the catalytic activity, whereas the zinc 2 plus has the regulatory activity. Please note that it is when MSH binds to the MLH1 that you have conformational changes that enhances the catalytic activity. So basically we are effectively understanding that MSH MLH is responsible for endonuclease activity, and also ATP binding and ATP hydrolysis is something that is responsible for ensuring that the endonuclease activity is active. If MSH is not bound, then the endonuclease activity of the MLH is not functional. So therefore, that is an extremely important fact. One another thing that is important to note is that the mute LH homolog or the mute LH system has to cut. So if it is an endonuclease, it is able to cut within the DNA. So if it has to cut within the DNA, Being the MMR machinery, it has to ensure that it cuts only the newly synthesized replicated DNA, because the wrong nucleotide that has been added on the newly replicated DNA. So the question that arises is, how does mute L homolog know which strand to cut? So, although the mechanism is not very clear, but there are a lot of evidences and lot of studies that have said that it is the interaction that the pCNA makes with the MSH. So, MSH, especially the MSH6, has what is called as pCNA interacting motif, and that pCNA interacting motif and allows the MSH to bind to pCNA. So in fact, you may consider that the pCNA is recruited wherever there is MSH, and interacting with pCNA already can be loaded with DNA pol, can be loaded with DNA ligase, can be loading or bound to FEN1. So therefore, the pCNA is somehow ensuring that the mute L orientation is such that it is only cleaving or forming the nick in the newly synthesized DNA and not the template strand. So that is extremely, extremely important, and that has been associated with the pCNA. So you can very clearly observe that you not just have MSH, MLH, but you have now MSH, MLH, DNA, pCNA complex being formed. Now what happens next is excision. And so we are going to basically understand how that excision happens. So let's say, for example, consider this is the mismatch, and well, the study has been done in plasmids where a mismatch has been created at a point, and then one has tried to observe in Saccharomyces how the mismatch repair system comes and functions. And like mentioned, it is able to recognize that strand as the newly synthesized strand that has the nick. So the nicks have been created. In this, it is created at the 5' side of the mismatch, and in this, it has been created at the 3' side of the um, mismatch. So therefore, you can have 5' MMR and you can have 3' MMR. But essentially, what happens is the mute S first comes and mute S alpha first comes and binds to the mismatch, and then it loads the mutal alpha. Now, the mutal alpha, when it binds to the mismatch, okay, what is observed is that you can have the mute SH alpha MLH PCNA RFC ATP dependent mechanism functioning in such a way that the mutal is able to create a nick towards the 3' end of the mismatch. So therefore, what this complex is ensuring is that there is a nick at both the sides of the, you can see, at both the sides of the mismatch. So there was already a cut at the 5' end. Now because of the mutal alpha mutes alpha complex, you also have a cut at the 3' end. So therefore, on either side of the mismatch, now you have a incision. Okay, now suppose you have the 3' cut over here and the mutes alpha and the mutal alpha come and bind, then you will find exactly the same happening. You will have an incision at the 5' end because of the mute L alpha, and so again either side of the mismatch you would have incisions. So effectively what you observe is you have an incision here, you have an incision here, you have an incision here, and you have an incision here as well. Now once that happens with the help of mutes alpha and with the help of pCNA, you have the exo1, which is a 5' 3' exonuclease coming and binding to the 5' cut, and on binding to the 5' cut, from here onwards it will excise out the nucleotides in the strand one after the other. So here therefore you have a gap being formed, and here also you have a gap being formed, and this is exclusively because of the exo1. So whether the cut is at the 5' end or the cut is at the 3' end, mutes alpha mutal alpha ensures that there is cut on either sides of the mismatch, And once the cut has been formed, it is exo 1 that will always bind to the 5 prime nick, and from that end it will then start removing the strand. So with that, you have the gap, and now the gap has to be filled up with the pCNA RFC DNA polymerase complex. So that is something that is going to happen. At this point, it is important to basically point out that there are several studies that have suggested that a 5 MMR can do without a mutal alpha, okay, but a 3 MMR cannot do without the mutal alpha. Primarily, what is basically understood is this, that from the 5 end, the 5 MMR, you can also have proteins like RPA, etc., playing pCNA playing an important role in excision with exo 1. So exo 1 can directly bind to the mutase alpha and do the needful. But in this case, there has to be a 5 cut created for the exo 1 to function. And so for the 3 MMR, you have to have the mutal alpha. A compromised mutal alpha is not going to help the 3 MMR, but a compromised mutal alpha for the 5 MMR is fine, because it seems there is another system available to basically ensure that excision happens. That's because exo 1 can directly bind to mutase alpha. So now therefore let us get an overview of 5 MMR and also an overview of 3 MMR. So you have the mismatch to which the mutase H comes and binds, and you have a 5 cut to which already an exo 1 comes and binds, but it is inactive. Okay, so and where there is a nick on the other strand, to protect the single strand over there, you have an RPA coming and binding. We know that RPA is basically equivalent to single strand binding protein, and it will go and bind to wherever you have single strand. Now, under this scenario, what is observed is that the mutase H, when it binds to the mismatch, due to the conformational change, it becomes a sliding clamp and it can slide. So it can either slide this way. If it slides this way, it may bind to a mutal H homolog present, okay? And once it binds over there, it may create an excision over here, okay? Now, secondly, what can happen, which means you must understand that it is not just one MSH that binds, you have several MSH that can bind to around the mismatch that is there in the DNA. You can have the MSH sliding towards the 5 nick that is there, and when it slides towards the 5 nick, it will bind to the exo 1, and on binding to the exo 1, the exo 1 will now become active and act as an exonuclease and start removing the strand. You see, it starts removing the strand. As it goes towards the mismatch, it will also remove the mismatch. Now, on the other strand, that is the template strand, you have the RPA coming and binding to protect it. So once that has been done, you have a pCNA that comes and binds with the DNA pol. It will start synthesizing the new DNA using the template, and this time as it synthesizes, it adds the correct nucleotide, and finally the sealing is done with ligase 1. Which is why, because you have the DNA polymerase, pCNA, the DNA ligase associated in MMR, you have to understand that the MMR is functioning closely with the machinery of the replication system. So this is important. So exo 1 on binding to MSH excises the strand, and pCNA with DNA polymerase and DNA ligase will fill the gap and also seal the gap. So this is how the 5 MMR can do. So you can see over here, even without the MLH, okay, you can have the excision being carried out, because exo 1 on binding to the MSH gets active and can basically carry out the excision, and then the pCNA along with the DNA polymerase fills up the gap. That is something that is noticed and observed. When you have the 3 MMR, okay, the 3 MMR, as you know, the cut is basically at the 3 end of the mismatch. So in this case, what is observed is that you can have multiple MLH coming and binding to regions around the mismatch. So, and you also have multiple rounds of MLH MSH insertion. You see, the MLH does not become an active endonuclease until MSH binds to it. So when you have the MSH sliding this towards the 3' nick and it encounters the MLH, see, without the MLH 3' MMR cannot happen. So when it binds to the MLH becomes active and it creates a nick. So you can see that now around the MLH MSH you have a nick here, you have a nick here. So you will have exo 1 coming and binding and removing this. You will have another MSH coming and binding and move towards this. So when it moves towards this, again it will bind to the MLH. It will activate the MLH's endonuclease activity, create a cut. The PCNA will come and bind to the MSH. The exo will come and bind. It will remove this. So when it removes it, the mismatch, the wrong nucleotide incorporated also is removed. So you can see right from here to here. So basically what is understood is several MSH and MLH are needed to create cuts within the region that is around the mismatch. And from those cuts, you have the exo 1 coming and binding and removing the DNA. So you have a large gap that is formed. The RPA is protecting the template strand. And then from this point on, you will have the RPA, you will have the RFC loading the PCNA and the DNA polymerase and carrying out the synthesis again, this time ensuring that a wrong nucleotide is not added, and then finally sealed with the ligase 1. So this is extremely important, and again this is a proof that the replication machinery works in tandem with the MSH MLH complex. So let's make the conclusions. MMR in eukaryotes is able to identify single base pair mismatches as well as 1 to 15 nucleotide long insertions and deletions. It also has been reported to be part of other molecular mechanisms like, for example, repairing oxidation-based DNA damage or also being involved in suppressing the homologous recombination. There are two families of MMR proteins that play a very important role in the eukaryotes, and they are the mut-S homolog and the mut-L homologs, with both being heterodimers and the nucleotide switch conformation enabling it to function optimally. MSH on identifying the mismatch complexes with MLH and PCNA to form nicks on either side of the mismatch, and then the exo 1 excises the strand along with the mismatch. PCNA, DNA polymerase, and the DNA ligase complex then fills up the gap and seals it such that the correct nucleotide is incorporated. Thus, MMR in eukaryotes, with its ability to identify the lesions in the DNA, are associated with the replication machinery to ensure that post-replication errors are corrected to maintain genomic stability. Thank you.